Hello, Mind Science, and welcome to our members-only BrainStorm event. And many of you have asked me some questions about, why are you doing this? What are you doing? Why am I here? Well, let me just, why am I here? So let me give you just a brief rundown of what we're planning to do, and then I'm going to turn it over to Marc, and he's going to amaze and delight you with our fabulous researcher scientists coming. So let me tell you, the genesis of this was a few things. First of all, we appreciate our members. That's each one of you. Thank you, thank you, thank you. You make it possible for us to fund research. There are researchers over here who have been and will be tonight the beneficiary of your generosity. And we thought, what better way than to bring the two sides together, the researchers and the donors, and give you a direct hand in choosing how we will fund this research. And I want to say something very special. Each one of these teams is a finalist. So just by being here and going through our very rigorous review process, each one of them, just by making it as a finalist, each has already won $15,000 for their research. Yes. Yes. So you're about to hear 3 stories of 3 different kinds of research. And what you get to do is everybody has your golden ticket. Everybody's got their vote that they can cast. And what we're going to do is after the presentations, I'll get back up here and I'll say, now go vote. And there are actual ballot boxes over on that side of the auditorium, and you will get to vote for your favorite project. And then Marc will come up and he will do some more amazing, I can't even tell you about it, it's a secret, but you'll see it. And while he's doing that, our crack staff of Emily Boyce and Jennifer Adams will be counting the votes, and then we will declare a winner. And at the very end of the evening, we'll get that wonderful moment of presenting that grand prize check for $30,000. So super excited. Thank you for being here. And the only other few things I want to say are thank you to our sponsors, Greg and Hedda Hahn, HAL Computing, and Kina Forgeson. Thank you to our board for support. Obviously to our members and our volunteers. So without further ado,
here it is. Thank you. Thanks, everybody. Thank you. And now on to our competition. Our first finalist hails from Northwestern University in beautiful Evanston, Illinois. His hobbies include playing keyboards, and in fact this weekend he's going to debut with a very special band of neuroscientists at the Society for Neuroscience. Tonight, he will give his presentation, and this is your cue. Are you ready? Please welcome, speaking on Inside the Mind of the Dreamer, Ken Paller.
I'd like to point out that Karen Kackley is my grad student. She's just started in my lab, and we're working on this together. She's going to lead this project. We're very excited about it. But before I tell you about it, I wanted to thank the Mind Science Foundation for the wonderful support of science they've been doing for many years, and thank all of you for keeping it going. I think it's just wonderful, and we're all very grateful that you've been doing this. It's very important. And so I'd like to invite you to be involved in our research a bit. And to start off, let me ask you this question. Have you ever wanted to be able to fly like Superman? You could have your arms outstretched, you could fly over San Antonio, you could fly somewhere else, you could go to the moon, anything you wanted if you had that ability. So one way to do that is to buy these virtual reality goggles. You put them on, it puts visual information into your brain, and that makes you think like you're actually flying. And maybe if you get a fan, you could blow the air through your face and you could feel it. But you can do all that. It's not really like flying like Superman. It's not the real thing. It's not even like dreaming it, because in fact, if you dream it, it's very real. That's one thing about our dreams, is they're the original virtual reality. When you have a dream, all the senses are there, and you really think it's happening to you. In fact, most of you, when you remember a dream, you have this experience of thinking it was real until the moment you wake up and realize, oh, actually, that was just a dream. But up until that point, you're accepting it as real, even when bizarre things happen. And the strangest things can happen, and you just keep going on with it, not sort of questioning, is it a dream or not? Well, there's a special type of dream that's called a lucid dream, and it's a rare event, but when people have that experience, they get the realization that they are dreaming during the dream, and they continue to dream, and they understand, actually, I'm sleeping somewhere, but right now, I'm having this experience of being somewhere else, and it's all manufactured by my brain, and in fact, I can control it if I understand that, and I can make anything I want happen. I can do anything. I can fly like Superman, and so on. And so that's an interesting phenomenon that we wanted to study, in addition to trying to understand dreams in general, because science has had a hard time studying dreams. It's been difficult. Dreams are something you learn about another person's dream only when they wake up, and they tell you about what happened. And we thought, well, we could do better than that, perhaps, by interviewing someone during a dream itself. If we could have a conversation with someone, then we could actually have a better way to study what's going on. So that's the idea. It's called a two-way communication between the experimenter and the dreamer. And someone comes to my lab, as shown here. They put on an electrode cap, and they're wired up, and they get to go to a comfortable room to have a nap. They lay down, they have a nap, and we record their brain activity like that, and we can monitor what's happening in their brain as this happens. And so we've been doing this for a while, but we haven't been studying dreams yet. That's our new adventure here. And so when we do this, we can look at this idea of two-way communication. But it really kind of seems like an impossible thing to do. How could you possibly communicate, have a conversation with someone we're dreaming? Well, in my lab, we have an answer. We like to say, well, we have a nap for that. In fact, we also have an app, because part of what we do is to use this app on a smartphone to make it work. And I'll tell you a little bit about how that works. Of course, there are a couple problems having the communication being both ways and sort of making someone have a lucid dream. It's not very easy to do. People have worked at that. It's very labor intensive, but we have kind of a shortcut that we're working on. And so the first part is, how can we figure out how to make this communication happen? How do we get the sleeper to tell us something? Because when you're asleep, your body is paralyzed. And so if you're in a dream, you're walking around, but in your bed, your body isn't moving. But there's an exception to that, which is your eyes. So when you move your eyes when you're asleep, your actual eyes move too. And so we can instruct people that when they want to signal us, they can signal with a code like left, right, left, right. And if they do that code, their actual eyes are moving, and we can register that in our recording. So here's a little segment of EEG recordings, and you can see in the circular area, there's this wiggle that's the left, right signal. So it's a very obvious signal. We can see that even during REM sleep and detect when someone's telling us something. And in fact, we can go into more advanced signals with greater numbers of left, rights and sort of have a little code that works out a conversation. So that's how they can communicate with us. And this has actually been done before. But the part that hasn't been done is, how do we communicate with them? And to explain that, I have to tell you about some of the research we've been doing in the past few years, where we're trying to understand how learning happens. And we think that when you learn something while you're awake, it doesn't just stick in your brain. In fact, you're going to forget it unless you rehearse it. Practice is very important. And we found that one of the ways to practice we do every night without knowing it, which is we rehearse information in our sleep. One of the things that sleep allows us to do is integrate the new memories we've gotten with the old memories and decide which are important to keep and so on. I've written about that in a few papers. One I wrote for a journal that's called Frontiers for Young Minds, and it's a great journal that's pitched for 8 to 14-year-olds. So it's science education, making science interesting for kids. So when I wrote my paper for that journal, I called it, Do House Elves Clean Your Brain While You Sleep? So this is, house elves are these creatures from Harry Potter's world, where the kids know all about that, and the creatures clean up everyone's house overnight, kind of invisibly, and you can make all the mess you want, and they clean it up. And so that's the idea here that, well, there aren't really house elves, but it's like that's happening because the brain is busy working through memories. Your brain is very active during sleep, and that's one of the things you're working through. And now in order to study it, we've been using a couple of interesting methods where we present soft sounds or words to people while they're asleep. And we've published that in many other papers besides this a funny one. So in standard journals, we have a lot of publications, including one that's going to be on the cover of Scientific American in the next issue. And the idea is, to understand how this works, we want to manipulate the processing of memories during sleep. And we can do that by playing sounds that remind people of something they just learned. And we know that that changes memory storage because, not just because we're looking at the EEG, but because when they wake up, they're better at remembering that information than other information. So we can really strengthen memories during sleep and sort of understand that normal process. So now we want to take that method, which we've been using entirely in slow-wave sleep, a different type of sleep. We want to move it over with this study with Karen to REM sleep and look at dreams. So that's the two-way interaction part. So this is a recording from one of our volunteers. We'll call her Linda. And we had recordings from different places on the head. And we played a sound. Once we noticed she was in REM sleep, we played a special sound that she heard. It wasn't very loud, so it didn't wake her up. But she knew that sound because it was on her smartphone app. And during the day, she heard that sound. And every time she heard it, she knew, well, I have to check and see is this a dream or am I awake right now? And she would get used to asking herself that question whenever she heard that sound. So she was kind of conditioned to make that check when she heard that sound. And then we did it in REM sleep, and she made the check, and she discovered, wait, actually, I'm in a dream right now, and I have to make that signal left, right, left, right. So about 4 or 5 seconds later, at time 2, she makes that signal. And so this is the beginning of two-way interactions. We want to ask more questions, get more answers, and have this be replicated in other people. So that's our project, is to try to use this method to move us forward so we can ask questions about what is sleep consciousness all about, and actually get some more information about how it works. So finally, to finish up, in addition to trying to study sleep consciousness in this way and understand how that might vary in different sleep stages and so forth, we also think there's some potential applications of our method. And so we might be able to use the same kind of strategy to help people. So one of the ideas is we can help people that have recurring nightmares sort of deal with their nightmares by using these sounds and inducing people to understand that they're dreaming at the time and then control what's happening. Maybe go ahead and talk to the monster that's bothering them and figuring out what that's about. Another idea is some people have very expert skills. They might be an expert musician or athlete, and they want to just get a little better so they can win the competition or something. So they might be able to use their dreams to practice their skills and rehearse it in the same environment, get a little bit better. Another idea we have is problem solving. So you might be struggling with a problem in your life and having trouble dealing with different people or something at work, and you could then visit that problem in your dream and think more creatively, perhaps, about another solution. So dreams are very thought to be a time where some creative solutions come, and we could provoke that to happen even more often to enhance creativity in that sense. A fourth idea is dealing with anxiety people might have. They're nervous about something, maybe they have a phobia. They can deal with that in the safe environment of a dream and work through the problem and maybe be better when they're awake and be better able to deal with that. And then lastly, of course, their imaginary experiences like flying you might want to have, or, well, whatever you want to do in your dreams. You could do that once you can control them better using our method to make this kind of dreaming happen. So that's the story. We want to try to understand what the mind of the dreamer is going on, and of course use our neuroscience methods to measure how that's happening. And sort of we think about this as kind of having a conversation with someone off in a distant world, maybe almost like the guy walking on the moon for the first time, and just having this conversation. We're in there in our lab, and we can talk to this person while they're dreaming and have this interaction, and who knows what questions. Maybe you have ideas for the questions we might want to ask them once we're able to do that. So thank you very much for your attention.
Good evening, everybody, and I'm really excited to be here today. I'm very thankful to the Mind Science Foundation for giving me this huge opportunity, and I'm also thankful to Dr. Mike Anderson, my supervisor, who is here to support me. So, and I'm very delighted to be explaining our project here today. So, if there ever was a law of human behavior which could be trusted, I think it is this. When people find themselves in an unpleasant situation, they immediately try to remove themselves from the situation. If your hand accidentally touches fire, you immediately pull it away. But what happens if this thing, which is harsh, is in fact stored in your memory? What happens if the pain is not being externally induced, but is emerging from the depths of your very own mind? It is a fact of life that most of us have experienced some painful, sometimes even traumatic incidents in the past. And when we confront reminders to such incidents, it becomes unpleasant, sometimes even scary. And what do most people tend to do in such situations? They tend to avoid the retrieval of such unwelcome thoughts and memories from intruding into their awareness. What we, in the memory control lab, we try to capture the essence of this very moment when people try to push the unwelcome thoughts and unwanted memories from seeping into their consciousness. I'm very interested in studying the brain mechanisms which are involved. And I'm also interested in this question. So what happens when this functionality fails? In order to illustrate this point, let me tell you a story. So this is the story of a man who was a great storyteller himself. Spalding Gray, as some of you might be aware, he was a great American writer, performer. He's famous for his autobiographical monologues, which he performed primarily on stage. And if you've seen his performances, he was very original and so full of life. But Gray's own story took a tragic turn when he met with a car accident. And as a result of the accident, he injured his right frontal lobe in his brain. And while this injury did not translate to an immediate loss of functionality, what it did lead to was a remarkable increase in the number of dark and depressive and ruminative thoughts that Gray started to entertain. And for years, Gray struggled to get away from these thoughts, but in the end, very sadly, Gray ended up taking his own life. All this led Oliver Sacks, the famous neurologist and a friend of Gray's, to remark, and I'm quoting him here, had some sort of a buffer, a protective inhibitory frontal lobe function been breached because of the accident, leading to an uncontrollable rush of previously suppressed and repressed thoughts and fantasies into his consciousness. Through our project, we intend to see if Oliver Sacks was correct or not. We intend to investigate if this critical region is indeed a critical shield in the brain. And in the process, we intend to study something vital about the regulation of consciousness itself. But how do you conceptualize the control of thought? If we think carefully about it, it is actually quite similar to one another form of control that we all indulge in on a daily basis. We are all involved in the stopping of our actions. We live in a world which is constantly changing, and in order to adapt to it, we need to be able to regulate and stop our actions properly. Could this stopping of actions have been the foundation for stopping of thoughts? Can there be similar brain regions which are involved in both forms of control? Do we know answers to these very important questions? What do we know so far? So this is a representation of the brain. And this is the highlighted regions that you see are the regions in the brain which are activated when people are engaged in stopping their thoughts or controlling their actions. This is a result from a meta-analysis study. So you're looking at a slide which has about 1,500 people's worth of data. And we are interested in this precise region of activity because results have shown that activity in this region is critical for when people are engaged in controlling their thoughts and their actions. But imaging studies can only tell us so much. They can only take us so far. What I think would be really cool is if we can put all this tickets knowledge to test, and if we can actually investigate and see if this critical region is indeed helping people to control their thoughts and their actions. And that is exactly what we propose to do in our project. But how do we go about proving the functionality of a brain region? It would be helpful if we can temporarily shut down activity in that region and see if that affects behavior. But can we really do that? It turns out now we can. So we have this innovative, fancy technological tool at our disposal now. So this is a non-invasive brain stimulation technique called transcranial magnetic stimulation, or TMS. TMS works on the principle of electromagnetic induction. The coil you see there is the TMS coil, and it's completely non-invasive, completely safe, and it's very focal, and it's placed on the scalp of the person you're stimulating. And we would be placing the coil in our critical region in the right frontal lobe. And TMS, it turns out, can be used for different purposes. It can be used to temporarily disrupt brain activity in the targeted region. It can also be used to temporarily enhance brain activity in the targeted region. So in the first project that we are proposing, we plan to disrupt activity in this critical region, in some sense simulating the Spalding-Gray experience, and seeing if that affects people's ability to control their thoughts and their actions. And if we are indeed successful in proving that this region is critical and it plays a shield function in the brain, then we propose to do something even better. We propose to use the very same technology, TMS, and see if we can actually enhance the functioning of this critical region. And this is very important because if you consider disorders like post-traumatic stress disorder or PTSD, which many people, including war veterans, people who've been through natural disasters or personal trauma, all suffer from, it's inherently an inability to control the painful thoughts and memories which keep seeping into their consciousness. Now, if you are able to enhance a critical region using non-invasive brain stimulation, we are actually making people better at controlling these unwanted thoughts and memories. Spalding-Gray's story does not have a happy ending. Gray ended up taking his own life. We need to remember that many Spalding-Grays live here amongst us. Some of you might even know of someone with a similar story. All these people are constantly fighting a battle which is emerging from the pain, which is emerging from the depths of their very own consciousness. Brain injuries, trauma, stroke can all lead to an impaired shield in the brain. Through a series of cleverly designed experiments, we intend to offer some insights into and also provide possible means of enhancing the functioning of this critical shield region, which we believe could be a crucial armor in people's struggles against the adversities of life. Thank you.
Thank you.
Thanks to the Mind Science Foundation for running such a wonderful program, and to all of you for coming here on this nice evening. So we've heard a lot about memory tonight, and memory ultimately has to be written into the brain at the level of molecules and cells, and people believe that your experiences are written into the brain by changing the strength of the connections between neurons, with changing the strength of synapses. So for your being here tonight, the memory of that is making some synapses in your brain stronger and others weaker, and memories are stored in big arrays of synapses throughout your brain. Now, what we would like to talk about tonight is not how memory is stored in the brain, but how your memories and your individuality are retained in the face of massive disruptions to your synapses. And let me give you a couple of examples. Men who have testicular cancer and who have to undergo surgical castration will see a 1/3 reduction in the density of synapses in their brain. It is then relieved upon testosterone replacement therapy, and the level of synapses will go back up. Similarly, women who have ovarian cancer and have to have surgical ovarectomy will also see about a 1/3 loss of synapses in the brain, and with estrogen replacement, we'll see it go back up. But the question then becomes, in these situations, memory is not lost. People are not rendered amnesiac by this treatment. You can blast a 1/3 hole in your array of synapses that appears to be random, grow synapses back in a way that may be entirely different than the way it was before, and yet our memories persist. And this is a fundamental puzzle. It's even more of a problem if you're a 13-line ground squirrel. Is anyone a 13-line ground squirrel? These are animals that hibernate, and when they hibernate, they lose about half of their synaptic density, and even the entire structure of the neuron is altered. Now, humans don't hibernate, but they do sometimes get a bit chilly.
Yes, so we're using hypothermia, the exposure to cold temperatures, as a tool to replicate partially what happens during hypothermia. This happens, for example, in humans during near drownings in cold water. And one of the best documented cases of this is of a Norwegian skier who fell into an icy creek. And she was pulled out from under the ice after 80 minutes, and she was not breathing, she did not have a pulse, and her body temperature was just about 57 degrees Fahrenheit. So essentially, she was clinically dead. And nevertheless, at the hospital, she was rewarmed and revived and ended up recovering without any lasting brain damage. Hypothermia is also used clinically after traumatic brain injury or stroke, or during certain open heart surgeries that require stopping circulation. So clinically, it's been shown that hypothermia is neuroprotective. And what I, as a basic scientist, find fascinating about this is to think of this in the following way. As the brain is cooled down, the electrical activity in the brain slowly slows down until it essentially stops. But nevertheless, the brain can then reboot almost as if nothing had happened and resume functioning just as it did before. So whatever makes us us can be preserved or frozen in time in a brain that is devoid of measurable electrical activity.
And so we would love to study this at the level of nerve cells and synapses in humans, but there are a couple of problems with that. First of all, there's an ethical problem. We don't want to make people hypothermic just to satisfy our curiosity. But the more important problem is a scientific problem, and that is the kind of brain scanning images that you've seen before here are very low resolution. So a brain scanning machine can show you features that are about 1 millimeter on edge, and a synapse is about 1,000-fold smaller than that, about a micron on edge. So our solution to this problem is to use our mammalian cousin, the mouse, and to, in particular, to use a genetically engineered mouse in which neurons in the brain have been caused to glow green in the dark. We then make a small hole in the skull and implant a piece of glass in the hole. And then, after recovery from the surgery, we can literally peer through that hole with a microscope through this window into the living brain of the mouse.
So I'm going to give you a short insight into the experiments we're doing. So we give our mice hypothermia. Here they're cooled down to 60 degrees Fahrenheit. This is actually a movie, but not much is happening because the mice are so cold. Here I am picking one of the mice up, and it normally would never let me do that. Only 2 hours after warming them back up, though, they're back to being normal mice and running around, going about their business. And this nicely recapitulates the sometimes rapid recovery that humans can undergo after accidental hypothermia. As David said, we can peer into the brain of living mice, and this is an example image. Just to orient you, this is about as wide as 2 human hairs, and these green tentacles that you see there are called dendrites. They're parts of neurons that spread out over wide areas and receive incoming information. And I want to show you a part of this dendrite in more detail. So these are 8 sequential images of that dendrite taken over a 5-week period, before, during, and after chilling the mouse down to 60 degrees Fahrenheit. Just to give you a little more information of what you're looking at here, there are these knobby protrusions that come out of the dendrite. They're called spines, and they're the locations where synapses are formed and where the dendrite, the neuron, receives incoming information. And these spines provide a wealth of information for us. As you can see, most of them remain stable over time, but some of them disappear, while others are newly formed. And some are transient, meaning they come up 1 week and then they decide not to stick around. So this reflects changes in the synaptic makeup of that neuron and probably underlies changes in information processing in this neuron. And we're interested to see whether chilling has an effect on the number of spines or the rate at which they're formed or eliminated. And this will tell us how chilling the brain affects information processing in the brain. And one last thing that we found, we're very excited about, is that these round swellings that you see there, those are not spines, but they're actually signs of injury. So it appears, it comes up 1 day after chilling. So it appears that chilling does cause a transient injury, which is intriguing when you look at it in that clinically, hypothermia has been shown to be neuroprotective, yet it also causes transient damage. So we're interested to explore further how this might inform down the road clinical management of therapeutic hypothermia. So we're very excited as these results are starting to come in, and we look forward to lots more experiments and data.
So it's remarkable that Julia's experiment can show us the very substrate of memory through this window. And I think it's worthwhile thinking about what the possible outcomes, the possible resolutions are of this puzzle. One idea is that you do lose a lot of synapses, and they come back, and they don't come back where they used to be, but memory is stored in just such a dispersed or holographic fashion that it can tolerate that amount of disruption. Another possibility is that maybe the synapses that are lost and come back are ones that don't participate in memory. Or another possibility that we're very excited about is that maybe when synapses go away, they don't really go away. There's a little seed that says when the warmth comes back, or in the case of ovariectomy, when the estrogen comes back, or in the case of castration, when the testosterone comes back, don't go randomly grow in this spot to reconstitute the information. So to us, this is very exciting as basic scientists because it is fundamental to how memory is stored in the brain. But as Julia has said, it also has clinical implications, so that when you go in for your aortic arch repair surgery or your aneurysm repair surgery and they stop your heart and they chill you, we hope that this kind of information will guide the parameters of that clinical practice in a way that will benefit all of our health. Thank you.
So while we are waiting for the votes to be counted, I would like to welcome back amazing misdirection expert straight from New York City, Mr. Marc Minton.
Thank you very much. So today I'm going to give you a very, very quick, as the votes are being tabulated, and our finalists are right there, we're going to do a quick talk on how magic works. Because I think it's really great. In a way, magic, some people have said it's like a defense of humanity itself. It's a defense of nature, of empiricism. And what I love is, let's hear it for Tom Slick. Okay? Because, exactly, and that's only appropriate for standing, because it truly is amazing. I think if he was here, which he might be in his spirit, is that what's so funny, it's so amazing, is that he would be so excited. And you can just feel it. He was a real lover of mystery, and actually inspired by Catherine Nixon Cook's beautiful book, The Mystery Hunter. So this was a man who was not afraid of mystery. And more these days, things like nuance and things like, talking through or disagreeing and accepting that. How many guys just have the guts, and women, have the guts to just get on a plane and go to Nepal and look for the Yeti, right? That's like a kind of embrace of other and mystery. And then I think of Catherine covered in leeches, following the path of, it's just amazing that you actually pursued that same kind of adventure. And so in that adventure, we can also be mystery hunters tonight. So as we explore magic for the next few minutes, then think of yourself as adventurers. Right? And so think of Uri Geller coming here, right, years ago. And actually, he's just moved. He is a friend. We've never discussed his stuff, but I've watched it very closely, and he's amazing. We first met in Toronto. He just moved all the way from, he was living in England, just outside of Reading, in a beautiful place. And when George Clooney moved next to him, then he thought it was time to sell his house. So then he'd always wanted to go back to Israel, his home, and he did. But he can do the most amazing thing with just a normal everyday object, and just like this, if you just go like this, you can see, it's kind of amazing that he had the ability to just make strange things happen like it was nothing, right? And like that, and you can see it like this, and then also like, this is the weirder one. Of course, this became a famous scene in The Matrix, right? But to think that there's a guy that thought about that and made that whole idea up, it's just so strange. Here, we'll give it as a gift to Miriam. Okay. Or no, wait, no, sorry, I'm going to give it to Catherine. Okay, so I'm just going to do one quick trick, really fast. Where are the two girls that I was talking to who understand all tricks? The two little girls, are they here or are they outside? They left? Well, tell them that I wanted to feature them, okay? Okay, but are you with them? Yes. Okay, good. Ma'am, could you just, if you would just say any card at all, okay? But change your mind a couple times to make it more dramatic. Because this is just like kind of a wraparound, quick effect. Oh, sure, change your mind out loud. That's even better. I wouldn't have thought of that, but that's great. Okay, good. So say a card and change your mind and say another one. And this is a situation that all of us are in when you communicate with people, right? It's a dynamic situation. So go ahead. So say something and say something else, just change your mind. And then you guys tell the jokes in your mind. Okay, good. All right, ready? Okay, that's going to be tough. Wait. Wait. But that's 3. Okay, I don't know if I can do it, but I'll try. Okay, so 2 of diamonds, 6 of spades, and what? Oh, I didn't. It's the 10 of clubs? Was that right? Okay, so we've got 3 cards, and we have a gentleman in a very beautiful coat. Could you come right up, sir, if you would? And we'll go like this. And what I want you to do is take these cards, and just to, I don't want to mess up the photos, but if you could stand right down there. And we're talking about the difference between, give him a little shuffle, and good, and yeah, perfect. Oh, okay. And if you could just step out a little back, we're talking about the difference between what you think in your mind and what you feel in your gut. So I want you to throw the cards right here against my gut, all right? Yeah, as hard as you can, but go back a little farther just to impress people with your incredible strength. All right. Oh, shoot. Okay. I got an extra one. I got a 6 of hearts, but so it's a total of 4. But what was the first card you said? 2 of diamonds, we got that. Good. Oh. Good. Ready? And 6 of spades or clubs? Good. I was worried there for a minute. Good, I was worried. Okay, and what was the last one? 10 of clubs, thank goodness. All right, here we go. Let's give him a big hand. Thank you. Thank you very, very much. Because if you do a trick right, you hear it immediately. People react. But it's easy to confuse these categories because they're ambiguous. So also, it's a lesson in subtlety and nuance, because actually, the presentation can tip the scale of, you know, what are the 2 words? Oh, yeah, effect and method, okay? So actually, before I even show you this, I'm going to let you feel it, right? With this simple experiment, because all of you have bags. Don't take them out yet. Wait, I want you to see this. The simple act of taking a ball and throwing it in the bag. Doesn't seem like much, right? But here's what's interesting. What happens if we throw the ball up and it still lands in the bag? Right, see, what's weird is we think that we need that trip to see it land in the bag. But what if you don't see that trip and it lands in the bag anyway? It's just not how we see, right? So it's interesting to eliminate all the information. So if we throw it up, oh, and you can see, you don't even need the ball. Do you see that? What's weird is that information is inside of you without it. Now you might think, oh, it's inside him, but it's not inside me. But let's test that right now. I'm saying that information to a fine degree is inside of you, how to throw and catch an invisible ball. So I'm going to show you this little trick, right? So take out your bag, right? And when you take out your bag, right, that's good. And we're just going to do this by ourselves at first, okay? Because I just want you to feel your own system. And I'm going to show you the secret, okay? So the effect is led by the action of this hand, and it's going to be completed by the method, which is being performed by this hand. You simply snap your fingers on the bag, and you'll see it'll look like the ball is landing. Okay? So none of you are fine mimes, but you can feel when it works and when it doesn't work. Right? Oh. Right? Right? So just try that. Just try to coordinate it for just like 10 seconds. Okay? And if you can't snap your fingers, it's about the timing. So throw it up and hit the back of the bag. Just throw it up, hit the back of the bag, and it's fine, right? For all the non-snappers, that's your technique. Okay, good? Did you get it? All right, we're good? Now wait, let's take a quick survey. Oh, look at that was a good catch. All right. You know what's funny? These glasses have been bothering me because actually, you know, we have a lot of assumptions, right? It's funny. Okay, I guess I didn't need them. All right, so I've got contacts. Okay. So, all right. What did you feel when you were doing that? Magical. Okay, anything else? Fun. What else? Caution? Fraudulence? I wanted to be sure of what you were saying, sir. I was a little scared. Okay, yes? Anything else?
Uncoordinated.
Okay, now, I have tried to use this phrase in every other way, but one day a person said it this way. Did anybody have a distinct feeling at some moment when you were trying this, I really suck at this? So here's one thing about sensory motor learning that everybody knows that does a sensory motor thing, like playing an instrument or playing basketball or driving a car or whatever. There's a point at which you go, you panic, right? But what does that mean? That you know the way it should feel, right? So did some of you also have the feeling, oh, that feels right? Did you have that feeling? So isn't that amazing? And did you ever throw an invisible ball before? No, I'm getting so excited. But but you've never thrown an invisible ball, but you felt when it was right. So that's what I'm saying is like and with this thing before, right? It's a kind of juggling. We don't think of it, but it's a kind of epistemological juggling. So, okay, I know this is a weird one because you can see the trick and the trick, because trick sometimes means effect and sometimes it means method, right? And it's just weird, right? Right? So you all know how it works, but isn't that a weird visual, right? Right? Oh, sorry. That's why they're called tumblers. Right, good. All right. Okay. So, all right. So now let's try some other fun little exercises. So with your bag, okay. Wait, okay. No, let's do it because this one is too much fun. We're gonna cross patterns. One more time, and this is actually a fan of Martin Gardner came up to me during the voting time, and this is another one. This is actually, he reminded me of this. I had done this as a child, but maybe some of you did it too. So you make up that little tube, right? And now what you do, you're going to cross information streams, right? Because what you're going to do is hold your hand up like this. Oh, there we go. That's Martin's illustration. And you're going to look through, oh man. Oh, but if your bag's crumpled, wait, straighten it out. First, make sure that you can see through it. I should have done this before we crumpled those bags. But can you make it into a little tube? You might have to make it bigger than I was making it. See if you can you see through it? Can you see through your, okay, if you need a new bag, just grab your neighbor's. Okay, now I finally got it. What you do is you put it right next to, and now here's the next trick. Look as far away. Do not look at your hand. Look as far away from your hand. And if you do it just right, it'll look like you're seeing a hole in your hand. Do you see it? Right? So do you see the way that we stitch together things in a beautiful, magical way? Right, and I love turning with it. Here's another one. Okay, and with a sheet of paper will work even better because you'll get a nice round circular hole. Next thing, okay, well, let's do the magic wiener. Look through, right, the famous old magic wiener. What's funny about this one, hold your fingers in front of you about an inch apart, and now look at the wall on the other side. Look right through me to the back wall. And do you see the little wiener floating between your fingers? Now, do empirical testing. Make the wiener small. By moving your fingers apart, make the wiener as long as you can. Okay, and now go up and down, and you'll see this kind of weird double effect. So it gets kind of weird, but you can see, and that's kind of what we do as magic too. We make the wiener look right. But don't quote me. Do not quote me, please. Okay, but you get the idea. Okay, here's another one. It's kind of fun at a party. This is called Aristotles Illusion. Aristotle really wrote this up, but it was actually written up by Greek philosophers before him. And that makes me think that this goes back much farther. Actually, it's my wife, Susanna, who's over there. Thank you for helping and thinking of this, okay? Cross, okay, this is a pairs exercise. Cross, okay, so designate one person as the crosser. So, okay, and one person crosses their fingers, okay? And, so it should be every other. So, sir, you can't both cross. Yeah, that's good. Okay, be magnanimous. And, okay, one crosser next to a non-crosser. And next, all the people with their fingers crossed, shut your eyes, because the next instruction I'm going to give secretly to all the other people that are watching me now. Okay, so all the people, so are your eyes shut, all finger crosser people? Now, the non-crossed people with their eyes open, this is what you're going to do to your neighbor. You're going to do this, be very quiet, and do this, as strange as that looks, right? Just do that. Go up and down, up and down, up and down, and now ask your friend, okay, and keep going a little bit more because it's just so strange, and ask your friend with their keep your eyes shut, and ask your friend how many fingers you're stroking them with.
How many fingers am I stroking them with?
1, 2.
Well, I would say 2.
It's only 1.
It's only 1.
Isn't that weird? How many people felt 2 distinct fingers? You can open your eyes now. How many crossers felt 2 fingers touching them and stroking them? Isn't that weird? All right. So now we're going to do the bags with social information. We're going to add judgment of others to this little process, right? Always a fun little step. All right? Okay. Next. So stand up, and now there's 2 of you with bags, but you only have 1 ball between you. Again, don't quote me on that. Okay, good. Okay. So take that ball and throw it back and forth and play toss with your neighbor. If there's 3 of you, you can do this with 1 ball between 3 of you. We're going to add a little music. You're going to have about 2 minutes to do this. Just make sure it gets right. Come on, David. You can do it. So basically, that's what I'm saying is the way that magic works, right? Did you all feel it? Right? And it's wild. And it is like real magic that's inside of all of us. And if you think about it, this is the way we used to play. It's so beautiful. It's actually kind of a lesson on screen time, right? Because we spend so much time in screens. It's just amazing when we really interact with each other, right? It's extraordinary. And now, please welcome back to our stage, Executive Director of the Mind Science Foundation, Miriam Good.
I would like to have our finalists come join me on stage, and Emily Boyce, if you would come join me. So this is what we've all been waiting for. I would like to take a look at our grand prize, $30,000 winner, Subbu Sankarasubramanian. You are the winner. Thank you. Thank you. And Dr. Ken Paller and Drs. Linden and Brill are our runners-up, and they each get $15,000. Thank you. You made this possible.